Polymerase chain reaction (PCR) it's an enzymatic technique that was discovered in early 80s by Dr. Karim Mullis, and it is used in vitro to exponentially amplify and determinate region of DNA. This technique is based on an ability of DNA polymerase to synthesize new strand of DNA complementary to a template strand by catalyzing in the addition of nucleotides into pre-existing 3' OH groups. There are several variations on the regular PCR techniques that serve in a specific application, such as the nested PCR that use two pairs of primers in order we obtain sequences that are structurally unavailable. RT-PCR, which uses reverse transcript enzyme to obtain ctDNA from mRNA, QRT-PCR, which can quantify the amplification of DNA using fluorescent probes, PCR multiplex, which can amplify different segments of DNA in one reaction using more specific primers. Hot start PCR, which does not allow the polymerase to start working until the end of the first cycle, since this cycle liberates the polymerase from an antibody that is designed to target it. Among other PCR variants are make this technique relevant in all areas of biotechnology. PCR takes place through cycles, each cycle consisting in 3 steps. 1. The DNA denaturalization at fusion high temperature to go from double-strand DNA to single-strand DNA. 2. The alignment of the pair of primers to its complementary sequence. And 3. The DNA synthesis through the DNTPS addition by polymerase enzyme.
Different solutions have to be added in order to obtain a PCR reaction. Since 3 reactions were going to be prepared for this experiment, we decided to obtain a master mix with all of the reagents and then add the different templates and controls to the different reactions. Here we show a table that contains all of the reagents used to prepare the master mix. For each reaction we added a volume of the master mix, and then we added the template and the negative and positive controls. Then we programmed the thermocycler and then we put in the mixtures. Afterwards, the samples were obtained and ran in a gel electrophoresis. Also, we analyzed the forward primer, its hairpin structure and its homodimer, the reverse primer, the hairpin structure for the reverse primer, and the homodimer for the reverse primer. And here we can see the heterodimer that can be formed. Here we can see the gel electrophoresis in buffer TBE 0.5x, and the gel was loaded with different samples and controls to see their pattern of migration. In the first lane we can see Axigent's 1 kilobase pair ladder DNA marker, 5 microliters that were loaded. In the second lane we can see the PCR product, or at least some of it, and it's expected to be around 400 base pairs, and we put 5 microliters of it. In the third lane, the negative control was loaded, which was water and the master mix to form 5 microliters. And in the fourth lane, we can see the positive control, that is the PCR product, the correct one, around 447 base pairs, and that was 5 microliters. And all of the gels were dyed using CyberSafe 0.3x, and the electrophoresis was ran at 120 volts for 45 minutes. When we ran the gel, we realized that the amplification hadn't been successful as expected, because we could barely see the band near the 500 base pair ladder, and we had to change the contrast of the image for it to be appreciated properly. We can also see leftover primers and DNTPs in the gel, and the negative control gel only contains primers and DNTPs, whereas the positive control shows the expected pattern of migration.
Conclusion. We can conclude that PCR is a highly powerful technology that is available to researchers for the development of science. It is one of the most important tools for molecular biology and genetic engineering nowadays. Due to its versatility, it has even more powerful variants. However, it is a very sensitive reaction that must be prepared carefully to avoid contamination and other sources of interference. After analyzing the results obtained, it can be said that the results were not as expected. This can be said because our DNA extraction sample was pure. This was considered with the purity standards of the absorption analysis by spectrophotometer. We can deduce that many of the interference factors involved in the protocols of DNA extraction and the PCR procedures were the ones that affected the PCR. Some of these interference were that the DNA samples when run in the electrophoresis gel showed it was contaminated with salts and proteins. If it's infected with salts, then the polymerase cannot run as smooth as it can, and it can produce small fragments, and by that, degradation of DNA.